All right, this next section is going to be nucleic acids. For this, you're going to want to be able to explain the differences between RNA and DNA and then summarize the role of ATP in cellular reactions. So each cell stores information for how the cell should behave, respond to the environment, and replicate. This information is ultimately going to be stored in the form of nucleic acids. Nucleic acids are simply polymers of nucleotides. So the nucleotide is the monomer form, nucleic acid is the polymer form. So what we have in the next couple of slides, or what we're going to talk about, is a sample of DNA. DNA is just one type of nucleic acid. It's going to be responsible for storing information, for information about replication and protein synthesis. It's going to contain all of the structures, all of the instructions for the activities essential to life, and also conduct chemical reactions. So here's the structure of a nucleotide. All nucleotides are going to have a phosphate head, a sugar, and a nitrogen-containing base. So shown here, we have a sample of DNA. DNA is going to be just one type of nucleic acid. It's going to store information on how to copy itself, and it's going to specify the order of amino acids in proteins. Ribonucleic acid RNA is going to have a lot of different types with a lot of different functions, depending on what their role is. But DNA's primary goal is to store information. RNA's primary goal is going to be to convert that information from DNA into proteins. Coenzymes can be nucleotides. They're going to help regulate enzyme action. They can either speed it up or slow it down. And then ATP is actually going to be responsible for storing energy. This is a question that I like to ask on either homework or quizzes, but which of the biomolecules have the primary function, or have as one of their functions the ability to store energy? That would be nucleic acids, lipids. Lipids are used for long-term energy storage, and carbohydrates, which are used for short-term energy storage. Proteins do not have any energy storage functions. Only nucleic acids, lipids, and carbohydrates. So here is just sort of a comparison between RNA and DNA. They do have two different structures. The D in DNA stands for deoxyribose. R in RNA stands for ribose. DNA has 4 bases: adenine, guanine, thymine, and cytosine. So you can kind of think of this as the alphabet of DNA contains 4 letters: A, D, G, T, and C. RNA does not have quite the same alphabet. RNA has adenine, guanine, and cytosine, but it does not have thymine. It instead contains a nucleotide base called uracil. DNA is going to be a double-stranded molecule, whereas RNA is a single-stranded molecule. Because of its double-strandedness, DNA will form that helix. RNA will only form a single strand, and it's not going to wind around itself. So here's our DNA structure and our RNA structure. The backbone of DNA and some forms of RNA is going to be an alternating phosphate-sugar backbone. The bases are going to project to one side of the backbone, and the nucleotides are commonly identified by their base, since that's the only component that's different with the nucleic acid. Like I said before, the DNA is going to be double-stranded. We're going to have 2 strands twisted around each other in a double helix, held together by, once again, what are these dotted bonds called? Hydrogen bonds. Hydrogen bonds are going to be responsible for holding our DNA together. We're going to use, our double helix is held together with those hydrogen bonds between those bases. And we're going to, in order to pair these up, we're going to use complementary base pairing, meaning that adenine will always bind to thymine and guanine will always bind to cytosine. And this is going to play a very important role later on when we're talking about DNA replication and synthesis. ATP, the last molecule of the nucleic acids, is ATP. ATP is adenosine triphosphate. It's a nucleotide composed of an adenine and a ribose. Together, those are referred to as adenosine, and 3 phosphate groups: ATP is a very high energy molecule due to the presence of the last two unstable phosphate bonds, which are very easily broken. So here we have 3 phosphates strung together. This last one in particular is incredibly unstable. It's like having 3 brothers all in a row. Terribly unstable. So what we're going to see is there's a lot of energy holding this bond together, and when it breaks, it's going to release a huge burst of energy. The hydrolysis of that terminal bond, that terminal phosphate bond, yields the molecule ADP, so adenosine diphosphate. Di meaning 2, tri meaning 3. So when we break ATP to ADP, that T becomes a D, that 3 becomes a 2. We get adenosine diphosphate, a simple phosphate head that has snapped off, an inorganic phosphate, and a whole bunch of energy. And that energy is amazing. That energy can be used for so many things. That hydrolysis of ATP molecule can be coupled with chemically unfavorable reactions in the cell to allow those reactions to proceed. So, for example, key steps in the synthesis of carbohydrates and proteins and muscle contraction and nerve conduction, all of those are going to require ATP. This is why we call ATP the energy, well, are all going to require energy, and that energy can be transferred in the form of ATP. Now, ATP is highly unstable. It's not really used for long-term energy storage, but it's more of an energy transfer unit. So, I don't know about you, but I like to think of the energy storage the different types of energy storage in terms of banking, because that's what works for me. So your long-term energy storage is going to be like your savings account. That's going to be the fat, the lipids. Your short-term energy storage is going to be your checking account, your short-term energy. Your instantaneous energy is going to be ATP. I don't know about anybody else, but I personally cannot hold on to cash. Since I had my very first job, I had a debit card. Every dollar I ever earned went into that bank account. So when I hold on to the money on my debit card really tightly, I do not like to spend it. Cash, on the other hand, cash is birthday money. Cash is gift money. Cash is meant to be spent. So I do not carry cash. I do not keep cash. I cannot hold on to it. It's not stable. It's not a stable way to store income for me. So if I ever need cash, I will go to my checking account. I will pull the cash out of my checking account and I will spend it. If I ever get cash and need to store it, I will drop that cash off at my bank, drop it into my savings checking account, and store it there. ATP works much the same way. You're going to get it just before you need it because it's not stable. It's not a stable way to save your money. It's not a stable way to save your energy. So what the body does is it stores the energy as glycogen or stores it as fat. And as you need it, it's going to break it down into glucose. That glucose is going to be broken down into ATP, and that ATP is going to be spent right away. So that's why we refer to ATP as the energy currency of the cell. Every reaction, and it doesn't matter, just like you can use cash for any type of transaction, you can use ATP for any type of energy needs, whether you need it to move the cell, to conduct for nerve conduction, if you need to drive molecules against their concentration gradient across the plasma membrane, if you need to move a molecule from one side of the cell to the other side of the cell, whatever reaction you have, any sort of reaction you have that has energy needs can be supplied through the production of ATP. So anyway, that's ATP. We do say it's short-term energy, but I want you to think of it as not really short-term energy, but like instantaneous energy. It's where we're going to convert. We're going to convert glucose into ATP, and then we're going to use ATP. We're going to convert solar energy into ATP, and then we're going to convert ATP into glucose. It's not really a long-term storage plan. All right. So that's it for this chapter. Make sure that you're able to describe the structure of a nucleotide, compare the structure of DNA and RNA, what impact do their differences have on their function, and explain how ATP is used as an energy carrier.